Let me hit record. Hi, my name is Queenie. I'm a third-year bioengineering major, and I have been researching in Dr. Huinan Lu's lab since fall 2019. This coming summer, I'll be researching at UCSD under Dr. Esther J. Kwan on nanosensors as neurodiagnostic tools. Now, to get started, I'll be presenting on the cytocompatibility of Yttria-stabilized zirconia ceramics for cranial window applications. This was research sponsored by UC Leeds, and I want to preface by saying I was not physically in the lab at the time of this experiment, and my contribution was mostly literature review and editing of the paper. So we'll mostly be going over background today and a bit of results to get the full picture of the experiment. So let's get started. Some deep background. So there are a couple common brain diseases that affect many people in the world, such as Alzheimer's, which is a form of dementia, Parkinson's, which is a central nervous system disorder that affects movement and can cause tremors, brain tumors, and chronic traumatic brain injuries. And the diagnosis, treatment, and medical care for patients have been increasing economic and labor costs in recent years. And long-term monitoring or treatment is required for many of these diseases and sometimes can require invasive surgery, such as a craniotomy. So what's a craniotomy? A craniotomy is a highly invasive surgical operation where a piece of the skull is removed for access to the brain and then replaced. And as you can see with this, removing an actual piece of your skull and then putting it back, there's a high risk of infection and other surgical risks, such as death. It's a pretty invasive surgery. So what can we do to make the surgery a little safer or make these diseases a little more treatable without this? So we looked into light-based diagnostic and treatment techniques, which is something that has been advancing in recent years. These are non-invasive diagnostic and therapeutic tools, and they basically use wavelengths of light in the region between deep blue and near infrared to look at biological tissue. And this is because those wavelengths have a unique photon energy range of 0.5 to 3 electron volts, which is great for interrogating biological tissue because any greater than that range would cause CC or CH bonds to dissociate, or ionization can occur where a molecule gains or loses electrons. And energy below that range will cause water absorption, which dominates the transmittance. So you're looking more at water absorption than actual light diagnostic and treatment techniques. So some examples of these diagnostic and treatment techniques are photodynamic therapy, which is clinically used in brain cancer treatment, or photobiomodulation, which is utilized to treat chronic traumatic brain injury. Now, the issue with this is the poor penetration depth of light, which basically means that the light can't go through as well as you want it to, due to static and dynamic effects of optical scattering and absorption by the inhomogeneous skull and scalp tissue, which basically means that your skull is not smooth, it's a rough surface. It's not the same thickness all around because it's bone that's shaped through growth. So that greatly limits the clinical use of the light-based treatment techniques. Now, what can we do to solve this issue then? So people proposed the Cranial Window project, also known as the Window to the Brain project. And this was proposed because some possible solutions to get optical access were either leaving the skull open or thinning the skull, neither of which are good for the long term. Your skull is supposed to protect your brain, and if you thin it or leave it open, that's not going to be great. So we use a cranial window, which is a transparent skull implant that can be used for light-based treatment. And as a biocompatible material, it needs to be compatible with human tissue. It needs to be able to withstand laser treatment, and it should be able to allow visual access for diagnosis and monitoring of diseases in the brain, which will get rid of the problem of having an inhomogeneous or too thick of a skull to use the treatment. And sometimes instead of this, we do polish the skull to thin it and make it homogeneous and slightly transparent. But at that point, that's also pretty dangerous, and it's better to just replace it with a solid piece that could help protect the brain as well. And so what materials are we looking into for this cranial window? So first we looked at polydimethylcytosine, PDMS. This is an elastomer, and it's not very good for protecting the brain because it's a very soft material, and the transparency is also not that great. So PDMS is off the table. And so glass, we looked into glass next. Glass has really great transparency, but the issue with it is too brittle. And look at any glass cup in your kitchen or anywhere, it will break pretty easily. So that's not going to be great for protecting the brain. And then we found YSC ceramics, which has been the most promising because it's pretty transparent. It allows that access to the brain and laser and light access. It has great mechanical properties. It's strong and durable, so good for protecting the brain, good as a replacement for a piece of the skull. And it's non-degradable, which is good for cytocompatibility, which is what we're measuring, and which means if it's compatible with cells, because within the skull there are cells floating around, or just brain cells in general. And so this will hopefully save lives and get rid of expensive treatments or invasive surgeries. And so here are our experimental steps. First, we obtained YSC ceramics, both from a commercially synthesized YSC ceramics and aerosol spray pyrolysis synthesized nanoparticles, which were made by a partner lab into disc. We polish these ceramics, and then we measure the water contact angle and surface roughness before and after polishing. We then sterilize it and apply the direct culture method to different YSC ceramics. Then we measure the adhesion density, spreading area, and aspect ratio of the cells adhered to the samples and on the culture plates that are surrounding the samples. And then we measure the pH value and yttria and zirconia concentrations in the post-culture media. And for the sake of time, we'll only be going over some of these results. So let's look into it. Materials and methods we have the blue boxes, which were the YSC ceramics prepared using commercial nanoparticles, and then the gray boxes are the ones using aerosol spray pyrolysis synthesized nanoparticles from a partner lab. Using the key, we have R, which means rough, P means pyrolysis, and P is nanoparticles. And then the numbers mean different yttria concentrations, because the higher the concentration of yttria, the more transparent the disc will be. But at some point, we do have to take away from that, and it will start to weaken the ceramic if the concentration is too high. So we want to look at what's the best concentration, what's the best combination of YSC discs that we can use to have a good amount of transparency and a pretty strong ceramic. And so the direct culture method is basically when we sterilize the YSC disc and put BMSC, bone marrow derived mesenchymal stem cells, directly onto the disc in the culture plate. We use BMSCs because they are stem cells and they differentiate into other cells. They're also the ones that are found inside the skull. After 24 hours, we collect the media for analysis and we fix and stain the cells that are surrounding the disc or on the disc. And so if we look at what direct and indirect contact means, direct contact would be the cells that are adhered directly on the disc, and indirect contact are the ones that are attached to the places surrounding the disc in the media plate. And so looking at results, first, we look at surface morphology. Surface morphology is basically what the surface of the disc look like, and using 3D microscopic images to really look at it closely. We look at the surface topography, so how rough these polished and unpolished discs are. And we did polish some of them to increase transparency, remember. So as we can see, in the top row would be after polishing, and in the bottom row would be, before polishing, so the rough samples. And if you look in the same column, these are discs with the same concentration of yttria. They're the same disc, but one is polished and one is not. And as you can see, the polished samples are significantly smoother than the non-polished samples. And if you see all of the top row, actually, you can see that there's a similar smoothness to all of them, no matter which concentration or how they were synthesized or how they were made. And then here is the quantitative representation of the last slide. And we can see here that all the disc samples got smoother as they were polished, looking at the branches, comparing, for example, on the left side, 3YSC and 3YSC rough. And then you can also see that all of the polished samples were about the same surface roughness, measured in micrometers on the y-axis. And as you can see, they are quantitatively about the same roughness as well. And now here are some representative fluorescent images of our cells. These are the BMSCs adhered on the YSC discs in both direct contact and indirect contact. So the BMSCs in different control and reference groups were looked at after a 24-hour direct culture, which was explained earlier. We used alexafluoro-4AA pallidin stains for the cell cytoskeleton, and it stained that green. And you can see how much the cell has spread. And if we look at this, the greater the spreading area, so the bigger the cell has spread out, the more happy they are in the environment they're growing in, which basically means they like where they're growing, they like the media, they like the disc or whatever's affecting them, and they want to spread out more and adhere. And then we use DAPI to stain the cell nuclei blue, which helps us see how many cells there are. And if we look at this scale bar, it's 100 micrometers for all the images. So we're looking at a really, really small area of our cell. And here's the quantitative representation of that last slide. And we can see that there's a difference between direct and indirect contact in terms of cell adhesion density. So the adhesion density in direct contact on the left side, figure A, is affected by surface morphology of the sample and composition of the sample, as well as hydrophilicity of the sample, whereas indirect contact in figure B on the right has been affected by the microenvironment, which, for example, would be ions released from the samples from the disc, such as yttria and zirconia ions. So in figure A on the right, in general, the commercially made 8YSC disc, as indicated by the red circle, has lower cell adhesion density than the other groups. And so that's something that we want to look at and investigate further. And in figure B, you can see that the cell adhesion density in the nanoparticles as circled has a lower adhesion density than the rest of the indirect contact samples. And that's most likely because nanoparticles have a higher specific surface area because they're so small. And once they reach a specific concentration, they will become toxic. So that's dose dependent. So basically, cells uptake nanoparticles into the cell, and a little bit is okay. But if we use a lot, which we did in this study to establish the worst case scenario, then it causes a problem because the nanoparticle concentration in the cell is too high, and that will decrease the cell adhesion density of the material. So in conclusion, the in vitro cell studies showed that the BMSCs adhered on the polished YSC disc had normal morphology, and that the ones adhered on the unpolished YSC disc showed less spreading than their polished counterparts of the same composition. So for example, 3YSC rough had less spreading area than 3YSC polished. And under direct contact, we had the commercially processed 8YSC disc, and the aerosol spray pyrolysis process disc had lower cell adhesion density than other groups on average. And under indirect contact conditions, we found that groups of YSC discs showed higher cell densities than the nanoparticle control groups, but they were similar to the control groups of titanium, glass, and cell only, which indicates that YSC discs have good cytocompatibility with BMSCs, meaning BMSCs like the discs, and it's perfectly fine if we use it as a cranial window. So this study was really important for us to screen promising transparent YSC discs for these cranial window applications, looking at composition, processing, microstructure, and cytocompatibility. And we will need further studies to determine the long-term transparency and biocompatibility of YSC discs, so in a longer time span than the 24 hours that we used. Here are my acknowledgments. I would like to thank Dr. Huinan Liu, my PI, Changlu Xu, my grad student mentor for the past 3 years, UC Leads for sponsoring this project, and Laura and Nancy, the coordinators of the program, Dr. Guillermo Aguilar, Dr. Javier Garay, and Dr. Lorenzo Mangolini, who were the co-PIs on this project, and NSF Pyre for sponsoring this project. Here are my references, and let me know if you have any questions. Yikes, sorry.